Okay, this is chapter 35 of the book, The Medical Reformation, and the topic here is what is cancer? How does cancer grow? It's kind of a long chapter, so I'm going to split it into a couple parts. It'll probably be an A, B, and C, chapter 35, A, B, and C. Okay, now first of all, when you talk about a disease, you have to ask yourself, what are you talking about? What is the disease? And the most important question with just about any disease is what causes the disease? Now, as obvious as that is, medical students are not taught that. They're not taught that, nor are residents or fellows typically. So for cancer, we're going to go through the understanding of cancer. There's two main theories of cancer. The first one is the SMT. SMT is somatic mutation theory, and that's what most people think causes cancer. That's the one that medical students are taught, that basically certain chemicals or virus infections or other mutagens, they can just damage DNA, and that leads to the cell growing out of control, and that causes cancer. But there's a couple reasons not to really believe that, or to think that that's certainly a rare cause of cancer rather than a common cause of cancer. Number one, if you just damage DNA, most of the time the cell would, like die, for example, or it would become dysfunctional. What I'm trying to say is, in order for cancer to grow, it's not one mutation. It's a whole bunch of mutations. It's an entire new way of life. And the likelihood of, a mutation is like taking an audio CD and scratching it with a paperclip. It almost always makes things worse and damages them. Versus cancer, a better way to think of it is that it's an entire new way of life for the cell. I'm going to go into that in a little later. I'll have better pictures of that. I'm also going to tell you that's why the metabolic theory of cancer makes a lot more sense. Metabolic theory of cancer, MTC, comes out of the work of Otto Warburg. He was a German biochemist, considered the greatest biochemist of the 1900s. He won the Nobel Prize in 1931. And what he found was if he took human tissue culture and he exposed it to hypoxia, meaning deficiency of oxygen, some of the cells would transform into cancer. And what's thought to be the mechanism is that the lack of oxygen was damaging to the mitochondria, injured the mitochondria, and that the cell reverted to a primitive pathway where it functioned on anaerobic glycolysis. And then you would say, well, how could a cell have that ability? It is thought that at the beginning of the world there was no oxygen in the atmosphere, and all the initial life forms were anaerobic, meaning functioning without oxygen, and that oxygen gradually became more available, but it was sometimes intermittent, and primitive life forms, cells, would have the ability to revert back to anaerobic glycolysis to survive. So the thought is that when the mitochondria is injured, that maybe these cells, what they're just doing is reverting back to anaerobic glycolysis, metabolism. This is why cancer cells, if you're running on anaerobic glycolysis, you make only like 2 ATP per glucose molecule compared to making about 36 when you've got the mitochondria functional. So 18 times more ATP, energy production. ATP is like a $20 bill in a cell. This is what the kind of money you use for energy for the cell to do work, to do everything it needs to do. Okay, so a cancer cell will take up 100 times, in the ballpark of 100 times more glucose than a regular cell. And so that's why you can detect cancer cells on a PET scan. A PET scan is a positron emission tomogram run on typically, fluorodeoxyglucose. Okay, so it's labeled glucose that you can see it taken up into the area of the cancer cell. All right, so what they think it is basically when cells are exposed to hypoxia, some of them die, whereas some of them, though, can transform into functioning like an anaerobic bacteria and then surviving. And but what I'm getting at, and the reason why this is a big deal, is because it's not just one mutation. It's a whole bunch of changes in gene transcription to support an anaerobic way of life. Okay? And so what this means is this is consistent with the cell makes like a volitional decision, if you will. We're going to try anaerobic metabolism. Now, the cell has to have the pathways, the capacity to do that. And that's, of course, an oversimplification. Everything I say is always going to be an oversimplification. It has to be. Nature is a billion times more complicated than we pretend that it is, okay? But we're able to understand things well enough to often get very good results. All right, so hypoxia induces a transition into running on anaerobic metabolism. So that initial event, the inciting event, is called initiation in cancer biology. And you really can't do much about initiation. You can prevent initiation in the future by keeping your cells well oxygenated. I mean, you can certainly dramatically reduce your risk of initiation of a cancer in the future by keeping your cells well oxygenated. And even Otto Warburg was aware of that, quite aware of that back in his day, and wrote about that. However, you can't change a cell that was previously injured years ago. And cancer, once this initiation event occurs, it can take a long, long time for it to grow into a clinically significant cancer. Okay, the next phase in cancer development is what is called tumor promotion. And this is really the most important phase, because tumor promotion is where you have a chance to change things. You can turn things around here. You got a good chance to do that. So T. Colin Campbell, in his studies, as he wrote about this in China Study and his other books, he found that if he stopped the animals, the rodents, from eating animal protein, he could often stop their tumors from growing in the liver, for example. Animal protein tends to be a tumor promoter because it has a different amino acid composition than does plant foods. It has more methionine, an essential amino acid that contains sulfur. It has more leucine, a branched chain amino acid that tends to be the rate-limiting step for activation of mTOR. mTOR means mammalian target of rapamycin. It's sometimes called mechanistic target of rapamycin. And it's a nutrient-sensing pathway, like a contractor getting ready to build a building, and it has to have all the building materials available before it can build. So the smart move, if you've got cancer, is try to minimize availability of those building materials, and with that in mind, hopefully slow down mTOR, telling the cell to replicate. Okay. Now, plant protein has less methionine and it has less leucine, and so it's thought to slow down mTOR. We'll get into that in more detail later, the nuance of that. Iron is also higher, let's say, in red meat. Iron is also higher in fortified foods, processed foods. Iron can be contributory to increased cancer risk because it can damage mitochondria. That's another useful way to think of it. To me, it seems that there's some that where it's well known that they increase cancer risk, but to me, it seems likely to think that anything that's damaging mitochondria is probably going to increase the risk of cancer. Okay, now I haven't gone through all the papers because there's over 50 mitochondrial inhibitors, and I would have to go through the papers on all of them, but I'm just saying, if I had cancer, I would want to avoid all these mitochondrial inhibitors. I would want my mitochondria functioning really well, because what seems to be the case with cancer is you're dealing with a competition between the normal cells and the cancer cell. And things that improve tissue oxygenation and cellular health tend to favor normal cells over the cancer cell. Okay, estrogenic chemicals are bad because for estrogen-sensitive tumors, which is typically breast cancer, endometrial cancer in the uterus, and also prostate cancer, you'd want to minimize them because they can potentially stimulate proliferation of some cells, some cancers, especially in that context. Okay, insulin tends to be part of the whole anabolic activation pathway related to mTOR activation. Insulin-like growth factor also is related to mTOR activation. You also want to optimize the functioning of your immune system, because it's your immune system that actually keeps cancer under control. It's the one that removes cells that have been released into the blood and prevents them from metastasizing, from being able to find a foothold in some other location. So what I'm basically saying here is the biggest part of the battle is lost or won at the level of tumor promotion. Now there's also, of course, the concept of metastatic disease, sort of the third phase of cancer. And the cancer cells are cells from the cancer primary tumor are released into the blood. They can spread through the lymphatics or they can spread through the blood vessels, and they will try to find a location at another place. Like, for example, imagine you have a liver cell and it's hypoxic and it becomes injured and it can no longer do the job of a liver cell to detox things, to manage blood glucose, to produce bile and release it into the biliary system. It takes too much energy to do that. So a regular cell is typically a worker, and its job is to participate as part of a multicellular organ system and do the job, let's say, of the liver if it's a liver cell, of the kidney if it's a kidney cell, or wherever it's located. Versus a cancer cell is more like that Johnny Paycheck song, "Take this job and shove it," where it just says, screw it, I don't have enough energy to do this work. I ain't doing it no more. And just try to replicate and then move on to find a new location to live as metastatic cancer. Okay, so that's why you really want to slow down replication, slow down mTOR. Okay, and you also want to do things that prevent metastatic cancer from getting a foothold. And so a big one is keep your immune system functioning well. And the way you keep your immune system functioning well is get your sleep, avoid caffeine, avoid unnecessary psychological stress, learn how to handle your psychological stress as well as possible. Okay, continuing hypoxia, lack of oxygen to the tumor, will increase all these vascular promotion factors like hypoxia inducible factor, vascular endothelial growth factor, and also proteinase that are going to break through the extracellular matrix and facilitate the cancer spread into the blood vessels. Okay, and abnormal angiogenesis, meaning the formation of bad blood vessels. Okay, the cancer cell will also pump acid into its surrounding extracellular matrix, the tumor milieu, the tumor microenvironment, because hypoxia and acidity favor the growth of the cancer over the surrounding milieu. And you see, by having this basic understanding of cancer, it tells you things you can do. Well, if acid favors cancer, I can try to make sure I have alkalinity. Well, what's relatively alkaline? Plants, okay, fruits and vegetables. All right, what's acidic? Meats, animal foods, okay, because they got more methionine, more cysteine. Those get metabolized as sulfuric acid as part of their metabolic breakdown, and that generates a mild metabolic acidosis, okay? That's bad. All right, let's see, what else? Oh, also, when the increased psychological stress causes increased platelet activation, and increased sodium can cause increased platelet activation. And what that means is the platelets become more sticky, more prone to clotting. And they can actually form almost like miniature clots around the outer surface of a cancer cell in the blood. And when they do this, they hide the cancer cell from the immune system and make it harder for the body to remove it. So that's another reason why if I had cancer, I would keep my dietary sodium real low. Okay, let's continue on here. So like I said, promotion phase is your best chance to stop cancer, but you can also keep on fighting all the way to the end. And a lot of people who've had metastatic cancer did all these things and had really long survival times. The most famous is Ruth Heidrich's, but there's a whole bunch of other ones. Let's see. Oils are tumor promoters. Vegetable oils, anything that causes leaky gut is going to make you have increased postprandial endotoxemia with bacterial endotoxins, including LPS from gram-negative bacteria, LTA from gram-positive bacteria, and that prothrombotic situation is going to lead to more hypoxia. It's going to favor cancer, so you don't want that. Okay, cancer itself tends to be a little bit prothrombotic. Okay. Estrogen chemicals are also obesogenic, and so things that favor diabetes and obesity and hypertension, and that means more atherosclerosis, they're pro-cancer, all right? T. Colin Campbell had noticed, too, a lot of these plant-based communities in China, when he wrote the book Chinatown, had much lower incidence of cancer. Caldwell-Esselstyn had talked about how back in the late 1950s in Japan, where they ate super low-fat diets, like, mostly white rice, they had incredibly low rates of prostate cancer. Okay, you need your immune system to remove cancer cells. Cortisol suppresses the immune system. Well, cortisol is increased by stress, sleep deprivation, and caffeine. Okay, here is a prokaryote, and pro means also before, like pre. Karyote means nut, and it really means in this context pre-nucleus, so cells before they had nuclei. The other thing, too, when you hear the word prokaryote, the first thing everybody's taught in school is it's a cell without a nucleus. It's a primitive cell, like a bacteria. But really, the most important thing of it is it's pre-mitochondria. It doesn't have mitochondria, okay? And it can run typically on anaerobic glycolysis, in its cytoplasm. Now, a eukaryote, eu means true, in the scientific sense here, and karyote means nut, so this is with a nucleus. But the big unique thing about a eukaryote is it has mitochondria. The vast majority of cellular energy comes from mitochondria. Okay, now here is a diagram of mitochondria. And a mitochondria was originally a bacteria. It's sort of the theory of endosymbiosis, whereby an original primitive bacteria ingested one of these early oxygen metabolizing bacteria that was a mitochondria, and they now are symbiotic, meaning they have a mutually beneficial relationship to each other. And so the mitochondria is inside the cell, and it's producing energy with oxygen, and then it gives that ATP to the cell. But the cell is doing it in a favor, provides it a safe location to live, and provides it with oxygen by its contact with the extracellular matrix, and of course thus with the capillaries. Now the outer mitochondrial membrane is out here. This green line that squiggles around is the inner mitochondrial membrane, and that's where electron transport occurs along this inner mitochondrial membrane. That is super important. The space between the inner mitochondrial membrane and the outer mitochondrial membrane, that is called the intramembranous space. And this yellow area is called the mitochondrial matrix. So you're going to have all these proton pumps along this inner mitochondrial membrane, and they're going to pump protons into the intramembranous space. And you're going to generate a big gradient. You're going to have tons and tons of protons in intramembranous space. And chemicals want to equalize their concentration gradients. They also want to equalize their electrical gradients. So H plus is a proton. It's a positively charged ion. And it really wants to get into the matrix, because the matrix is going to have a negative charge of about negative 160 millivolts. So the positively charged hydrogen proton dramatically wants to get in there. It also wants to get in there because of its concentration gradient. It's very high in concentration in the intramembranous space, very low in the matrix, so it wants to come across. And the energy of that hydrogen proton coming back into the matrix through the ATP synthase is coupled to having the ATP synthase complex number 5, be able to add a phosphate to an adenosine diphosphate and make it an adenosine triphosphate. So it went from ADP with 2 phosphates to ATP with 3 phosphates. Okay, that's how it works. All right, here's a close-up look at the inner mitochondrial membrane. Here is complex 1, complex 2, complex 3, complex 4, complex 5. Complex 1 goes right across the inner mitochondrial membrane. This, by the way, is the outer mitochondrial membrane, and the space in between with the intramembranous space. Okay, here's complex 2, which is also part of the Krebs cycle series of chemical reactions. Here's coenzyme Q. Okay, you probably heard of that one a lot. Complex 3, cytochrome C on the outer part of the inner mitochondrial membrane. Complex 4, and that's a real important one, and oxygen accepting the 4 electrons occurs right about here, and it becomes water. And then here is ADP, gets a phosphate added to become ATP. This complex 5 is ATP synthase. Okay, so that's really key stuff. And I know this is kind of complex. If it's the first time you've heard it, it seems kind of complex. But I can tell you all life on Earth depends on this. The health of all your cells depends on this. And the amount of complexity I just described you is about all you need to know. It's like rolling a snowball down a hill. This has a relatively weak grabbing ability of electrons. And then each complex, as you move distal, has a more stronger ability and desire to grab electrons. So that's why the electron movement is favored to go from complex 1 to complex 4. And basically what inner mitochondrial membrane electron transport is, it's like a fireman bucket brigade. You know, they're passing the electron from complex 1 to complex 2 to coenzyme Q to 3 to cytochrome C to complex 4. Okay, and you can almost think of it as when you eat food, your food is basically being digested in stepwise fashion, where the electrons from the food, from the glucose, are being ripped off, stolen, if you will, by the cell and used to generate energy. Because you want to think about it, the cell is a great genius. What the cell figured out how to split the atom, you know, many millions of years ago, whenever it first developed mitochondrial function inside a cell. And so when I say split the atom, what they do is they split the hydrogen atom. They take the proton part of it, H+, and they pump that into the intramembranous space. Here's an H+. And they take the electron part of it, and they pass that along these complexes and use the energy obtained by letting it, in a sense, like a snowball rolling downhill. They use that energy to pump protons into the intramembranous space, and then that built up electrochemical gradient is harvested by letting a hydrogen come back through ATP synthase, and that energy is used to make ADP to convert it into ATP. That's how it works. That's where energy comes from on life. It's pretty cool. It's rather extraordinary. Okay, I'll show you one more diagram of it, and I think we'll call it a day at this point because it's going to get kind of complex. But let me just show you. But it's actually going to get simpler. What I just showed you about the electron transport chain, that's as complicated as it's going to get. But I just want to have you understand that. And if you look at any other pictures of it in other books or lectures, you'll see that what I just told you was all you need to know. There's always more information, but this is all you need to know. Okay, complex 1 pumps a proton out into the intramembranous space. Complex 3 pumps a proton out. Complex 4 pumps a proton out. Coenzyme Q is like a carrier. All right. And again, this is what I meant by splitting the atom. The electrons are passed downhill to progressively stronger electron grabbers. Oxygen is the strongest electron grabber. It has the highest electronegativity amongst these elements and complexes, so it just grabs that electron, and it gets in the form of the hydrogens as well, and it becomes H2O. All right, and then it's just a safe thing. Simultaneously, these are the H pluses, the protons that are pumped out from each glucose, and that's how energy on life is made. That's how most of your ATP is made for energy. So again, this is the IMM, intramitochondrial membrane. This is the IMS, intramembranous space between the inner and the outer mitochondrial membrane here. And then this is the matrix where Krebs cycle occurs, and complex 2 is part of Krebs cycle. Okay, so that's it for chapter 35A. Introduction to cancer metabolism, and we just covered electron transport in the mitochondria. Hope that's helpful.